I am optimistic that we will have a vaccine by December because we have new technologies available to us that were not available as early as a year or 2 years ago. And by relying on that new technology, we will be able to produce the vaccine quickly and in large numbers. And that new technology is something that will be used for the first time. So we don't have a long history, so people are surprised by the time frame of developing the vaccine, but we have the technology to do it. The technology relies on RNA, and so I need to explain that a little bit. So everybody understands DNA. DNA is our biological template, and it's, well, everybody understands what that is. Well, DNA is really like the specifications, the blueprint for a building. That is our blueprint. And like a blueprint, it needs to be read and interpreted. Well, RNA does that. RNA reads the DNA and interprets it, and from that interpretation creates the proteins that we need to survive and live. So unlike any other vaccine before, what the new technology does is, oh, and I have to say that the coronavirus is an RNA virus, that instead of DNA, it actually has RNA. And when it infects a cell, the RNA then uses the cellular your cellular machinery to create more viruses. So what the new technology does is it takes a small section of the virus's RNA and then inserts it into these small lipid capsules, little mini microcells, artificial cells, which are then inserted into your body. And your cells take up those microcells. And then once that messenger RNA is inside your cell, it takes over that little snippet of RNA, not the entire virus, but just a little tiny section of the virus's RNA. Your cell is then commanded to make the proteins, the exterior protein spikes that the virus has. And so you produce in your own body all of these protein spikes, which are then either introduced into the surface of the cell or sloughed off into the bloodstream. And so your body now has these huge targets, these amazing targets to produce antibodies against the virus, even though you don't have the virus. You just have the spikes that are presented to you when you do. So it's safe, and your own body is producing the spikes. And so that's another safety feature. Virtually every vaccine that's ever been made up until this date has used a method that's been around for 70 years. And it requires that the proteins that will be used in the vaccine or the attenuated virus itself actually are incubated in fertilized chicken eggs. It's a very time-consuming, slow, and expensive process. It works, but it's time-consuming, slow, and expensive. What we're talking about now is completely different. It's called recombinant virus. And recombinant is just a fancy name for our ability to manipulate the genome. We can manipulate DNA and RNA now in amazing ways. And unlike a conventional virus that has to incubate the proteins to develop and grow them, we can actually produce the genome segments in bacteria. And everybody knows that bacteria can produce very quickly. Some divide every 20 minutes. So in a very short, double in every 20 minutes. So in a very, very short time, if you can introduce these segments of DNA to be reproduced in bacteria, you can produce tons of this material in a very, very short period of time. So any drug, including vaccines, before being approved has to go through rigorous testing. And there are 3 post-preclinical phases, phase 1, 2, and 3. And phase 1 just demonstrates that it won't kill you in small doses. Phase 2 demonstrates that you can give it in reasonably therapeutic doses without significant side effects. And phase 3 actually looks at, in a large population, does it work? And is it safe? And does it have bad side effects? You need drugs to test to do those phases 1, 2, and 3. with a recombinant virus, with a recombinant vaccine like we're talking about right now, you can develop a large amount of material quickly so that you can do those tests quickly. That's another reason why I'm optimistic about December, because we will have the vaccines available for testing in large quantities because it's a recombinant vaccine. So phase 3 is what most people think of when we think about drug testing. It takes a large population of people, in some cases the people who have a disease like cancer, and we look to see giving a drug to that patient population if they either get cured or extend their life or in some ways help them. So with a vaccine, you're looking for whether or not in a large population, if getting that vaccine protects you against getting what disease you're vaccinating against. So you need a large number of people for the statistics of it. You need to prove that over a large group of people that the drug works, that the vaccine works. And you need a large group in order to do that convincingly. In addition, a large group will also yield more side effects because sometimes a very, very small subpopulation will react in a very different way than the majority of people react to any drug, including a vaccine. So you need a large number of people so that you can detect if there are subgroups that will react differently or badly. That's why you need a lot of people. Depends on the disease and the drug. In a vaccine trial, you're usually talking about somewhere between 20 and 30,000 people to do the study. Well, once you have a vaccine, however you develop it doesn't really matter. So the vaccine, unlike some diseases like cancer, which have long time periods between developing the disease and having some horrible consequences of it, with the vaccine against the disease, the endpoint is very quick. It's easy to detect whether or not it works or not. You give someone a vaccine and you challenge the population with the virus and you see if they get sick or not. It's very straightforward and it's very quick, unlike some other diseases. Yeah, we don't produce the cells, we produce the proteins. Our cells produce the proteins that the virus makes, but not the virus itself. So the virus, a virus is a little capsule of genetic material. And so you have, let's say, DNA or RNA, take your pick, because there are both types of viruses. So then you have a genome, just like we have a genome. It's the template for the virus. Then it's surrounded by a protein coat. It's its little capsule that encapsulates the genome, the DNA or RNA. And then that protein coat itself has, in the case of coronavirus, protein spikes that stick out of it, like an old landmine from World War II. So you've got the DNA, which is encapsulated in a ball of protein, a little hollow ball of protein, and on the surface of that ball of protein are protein spikes. That's what the virus looks like. And the immune system is really generally only seeing those spikes. So if you can make those spikes, the immune system will react to them. Even if there's actually no virus, just the spikes from the surface of the virus. And that is what that vaccine is doing. That's why it's so effective. You are hearing about long development times for the vaccine because the experts that you're listening to right now are thinking conventionally. They are considering the conventional means of producing a vaccine, including the time spent in incubation in fertilized chicken eggs. What we're talking about is something entirely new. And the time frame for development is much more rapid than the conventional means. And that's why there's a disconnect between what's really happening and what some of the experts are saying. The idea that we will be using RNA to develop the vaccine has consequences for the type of immunity that we're talking about. one of the beneficial side consequences of this new technology is that the vaccine will very likely induce T-cell immunity, and that will result in long-term, probably lifetime immunity against the virus. So the answer about what T-cell immunity is really is beyond the scope of this discussion, but we can touch on it. And there are broadly two types of immunity that we have. We have innate immunity and adaptive immunity. And when we generally think about immunity, most of us are thinking about adaptive, and that includes T cells. So what happens is when you're presented with an infection, your thymus trains these cells that are floating around in your body, very specific types of immune cells called T cells, to recognize the invader. And those T cells are lifetime. They will always remember for the remainder of your life that disease. And every time you are presented with that disease, your T cells are primed and ready to go and will attack it before it can make you sick. So any idea that we won't have long-term immunity is not based on fact at this point. There are no studies that show or indicate that the antibodies will not provide immunity. That's just speculation that you see on the internet right now. There's really no reason to believe it. I mean, anything is possible in biology. That's a remote possibility. But more likely, especially if we use an RNA virus, we'll have the type of immunity that you get when you get a vaccine against the measles or typhus or any of the longer term immunities that we get from our typical vaccines. So the type A influenza virus and coronavirus are quite different biologically. The flu virus briefly presents to the world two different primary proteins. One is neuraminidase and the other is hemagglutinin, the H and the N in H1N1, for example. Those proteins mutate rapidly and often. And that's why every year you need a new flu vaccine, because the different combinations of H and N are derived every year. That's not the case for the coronavirus. The protein spikes in the coronavirus are relatively stable, in contrast to the type A influenza virus. There are mutations. There's, in fact, 4 varieties of the coronavirus CoV-2 circulating in the globe right now. So there is mutation, but unlike in the influenza virus, in the coronavirus, those mutations do not tend to change significantly the protein spikes against which your immune system is reacting. Like anybody, I'm always concerned with the safety of anything that is injected into my body. I'm as concerned as anybody about safety. I am also confident, though, that if the vaccine is approved, it will have been proven safe through all the normal rigorous processes that we use to evaluate whether a drug is safe or not. In terms of whether it's effective, I answer it in the exact same way. The rigorous approval process will demonstrate whether it's effective or not. If it's not effective and it's not safe, it won't be approved. So the Russia claim about a vaccine is intriguing and interesting. The problem is we don't know enough about it to draw any conclusions. Perhaps it's a real vaccine, but one thing that we know is it certainly did not go through what we consider phase 3 clinical studies here. And so those questions of safety and efficacy have not been answered, at least as far as I can tell, have not been answered to the kind of satisfactory degree that we would expect here in the United States. Well, when I say that a vaccine will be ready in December, I mean ready for distribution so that people like you will have it. I think it will be available and ready and approved and safe and effective by December. And again, I draw that conclusion going back to some of the very first questions you asked. It's the way we're producing the vaccine using new technologies that we've never had before. I want to also make another point about that. Because we're producing it quickly and more rapidly than ever before doesn't mean we're rushing it. Doesn't mean we're rushing through the process. The process of determining efficacy and safety will be as rigorous as ever, if not more so, given the incredible scrutiny that it will have. It's only being done quickly because we can do it quickly given new technology. We're not rushing through this.
Yes.
So the process of developing a therapy against the disease is the same process for getting approval for a new drug. And it has to go through the different phases of phase 1, 2, and 3 after all the preclinical. That process is equally rigorous. And I would never even consider taking any therapeutic that had not undergone that very rigorous review process. It's crazy to take unapproved drugs or even approved drugs for unapproved diseases. I mean, that are not approved for a particular disease. And there are many reasons why that review process is rigorous. And you don't want to take therapies that are not approved. They can be quite dangerous. Well, the concept of herd immunity is generally misunderstood, but you're right around there. We don't really know what it is. In some diseases, it's as low as 60%, and with other diseases, it's as high as 75 or 80%. 70% is a good target. And what that means very simply is that if a large enough people are immune to the virus no longer has a reservoir in which to reproduce and spread and eventually will die. That's what herd immunity is. It's a simple concept. And so you need either 70% of the population to have been infected or been vaccinated to create that type of herd immunity. Even if 30% of the people of the population refuse to be vaccinated, which is a whole nother discussion, enough people will have already had the disease so that the remaining 70% who will get vaccinated may be enough to create herd immunity in spite of the ignorance of those who decide not to get it.